Hello students, welcome back to class. Welcome to Biology with Omotayo. We will be continuing from where we stopped with regards to the last subtopic under consideration during our last class. The topic or subtopic under consideration was cell structures and function. A bit of introduction was brought on board last class, where summarily the cell was being defined as protoplasm surrounded by a membrane. The protoplasm regarded as the living component of the cell. For more in-depth knowledge with regards to what was discussed, kindly visit last class video to acquire the required knowledge. For today, we will continue from where we stopped. So we'll be looking at that subtopic now in detail. Structures and functions of cell components. The structures and the functions of cell components. Also from that last class, emphasis were laid on eukaryotic cells. It was said in that very class that our emphasis will be on plants and animal cell. Even though we have more than two kingdoms, not just Animalia and Plantae, we have others, Monera, Protoctista, and fungi, but this study of cell and its structures and their functions will be limited to plants and animal cell. So, a quick one, I will illustrate the structure, or permit me to say, I will illustrate or draw the diagram of the animal cell and then identify some of the organelles, after which the functions of the labeled organelles will now be highlighted. Come along with me as we do this together. Class, I have just illustrated the diagram of an animal cell, and you can see that this cell is irregular in shape. It has no definite shape. That is simply because the animal cell is bounded by a membrane called the plasma membrane. The animal cell is bounded by a plasma membrane, hence it is not a rigid membrane, and so a shape is not retained. So the animal cell appears irregular and saggy. And so a quick one, together we'll be naming some of the components of this cell. As you can see, a couple of structures have been indicated in the cytoplasm of this cell. The cytoplasm is the fluid portion of the cell where organelles are suspended, and as you can see, the cytoplasm of our animal cell here is dense with several organelles located in it. Come along with me as we label this animal cell together. and we'll do that in a systematic way. Take note, in biology, when you are to label, we label left and right, or to the left or just to the right. That is to say, we label horizontally, and we equally label making use of our ruler in the drawing of our lines. In biology, you don't draw your lines with free hand. Always make use of your ruler so that you can get the mark allotted for neatness of labeling. You label with the help of a ruler in drawing out your lines, and you ensure your components, your labeled parts, are horizontally listed, either all to the right, all to the left, or both ways, left and right, but you maintain a horizontal part. And that is exactly what I'll be doing now. Come along with me as you watch me label components of this plant animal cell. Okay, class, as you can see, the diagram is now partly labeled, not fully labeled, but because of space, I've limited it to these few that have been labeled. So you can see that the nucleus alone has almost about 4 components. Okay, the chromosome, the nuclear membrane, that is the covering here, the nucleolus, the nuclear pore. All these 4 gives us the nucleus. Okay, and then we have the cytoplasm, which is the fluid portion of the cell. Then the Golgi body. We will later be looking at the function of each of these structure, the glycogen droplets, the mitochondrion, for singular, mitochondrion, please, not dria. We have the vacuole, tiny vacuoles all around our cells. We have the cell membrane, and another key component left out. We equally have the ribosome among several others. After now, the diagram of the plant cell as well will be drawn. The components will be identified, and then we will move on to look at the function of the various structures identified in both the animal cell and the plant cell. Please take note, for the animal cell here, you can see that the nucleus is almost centralized, and we have vacuoles that are small, okay, and numerous. When we get to plant cell, you will decide and come to see if the same will be the case for the nucleus of our plant cell and if same will be the case with regards to vacuole in our plant cell. Come along with me as I illustrate the diagram of the plant cell as well. Okay, class. So I will now be illustrating the diagram of the plant cell. Come along with me and try do the same. Okay, class, this is my diagram of the plant cell. I will equally write my title. And then together we'll be labeling this diagram. Just like I did in the earlier diagram, I will do a similar here by beginning to label my nucleus and the components that makes up my nucleus. I've been able to do a bit of justice to this diagram with regards to labeling, and you can see that a couple of components have been labeled here. All in all, our nucleus has about 4 components coming together, and from there we went on to look at the smooth endoplasmic reticulum. It has no ribosome attached to it, so it is called the smooth, while the endoplasmic reticulum that has ribosomes attached to it, usually found and clustered around the nucleus, are called the rough endoplasmic reticulum. You can see for yourself, just as I have asked you to watch on from the last diagram we drew, that the vacuole here is much larger, far larger, and the nucleus and its components are not centralized in the plant cell. Rather, it had been compressed to one side of the cell. Also, as you can see here, we don't have just a cell membrane, we also have a cell wall. A cell wall was not labeled under the animal cell, which was the reason why our animal cell appeared irregular. It lacks a wall. But for the plant cell, you will see that a defined shape is obtained because of the presence of a cell wall. Also, under the animal cell, we labeled glycogen granules. But as you can see, under the plant cell here, we rather have labeled starch granules. Why? Because glucose is stored in animal in the form of glycogen, while glucose is stored in plants in the form of starch. And as you can see, we equally have the oil droplets here. The Golgi body, or what we usually refer to as the Golgi apparatus, is another organelles here that serve the function of packaging and distribution. This will be seen when we come to look at the basic functions of these various organelles in the cell, and then our cytoplasm and the mitochondrion, that is singular of mitochondria, an organelle which is being nicknamed as the powerhouse of the cell because it carries out respiratory activities. Students, I believe with these two illustrations, the earlier one, that of the animal cell, and this very one, that of the plant cell, you have come to appreciate better the structure of cell, with emphasis on that of the kingdom Animalia and Plantae. See you next class, where we will begin to look at the various functions of these structures that have been identified here, their uniqueness, and how they help the cell to function and be able to carry out its unique activity, either as independent cell, as group of cells such as colony or filaments, or as a component of a multicellular organism. Please do well to revisit this video and study privately on your own again. Kindly endeavor to like this video, endeavor to share it, endeavor to inform others about this channel, so that together we can learn and collectively build a well-informed global community. See you next class. Thank you for joining this far with Biology with Omotayo. Take care.